Organogenesis. Now genesis means formation, organo means organs. Organs formation. Now we are taking first, that is here we are taking the callus. Okay, the callus. Imagine that the callus is divided into small pieces. One piece of callus we are taking. It undergoes differentiation. What is the meaning of differentiation? It is becoming stimulated or induced meristem. It is changed into permanent tissue. Permanent tissue means parenchyma, collenchyma, sclerenchyma, like that. That is called a differentiation. One side they are differentiated into shoot system. What is the shoot system? Above the soil, the main stem, branches, nodes, internodes, leaves, buds, flowers, fruits, everything together we can call as shoots. The development of shoot from the callus is called caulogenesis. Caulo, C-A-U-L-O. Caulo means a shoot. Okay, genesis means development of the shoots. Then at the time of differentiation, one part of the same callus is developing into roots. Okay, they are developing into roots. That means what? Primary root, secondary root, tertiary root, root has everything. That is called the roots. It is called rhizogenesis. Rhizo, roots. Rhizo is other term for roots. Genesis, development. Development of roots, rhizogenesis. Now this is second. So the shoot, entire shoot, what I told, above the soil, what are the things are present, and below the soil, roots will be present. Everything together we can call it as plantlet. A tiny plant, a complete plant. That is a basic concept of organogenesis. Now we are coming to applications of tissue culture. Okay, we have developed the technique. The scientists are doing a lot of research and they are spending a lot of time and they are discovering this tissue culture technique. Then why we have to do that? Why we have to discover this tissue culture? It is called application. Now we have to apply the technique in various plants so that we must be benefited. Okay, we must be benefited. Human beings must be benefited. That is called the applications of tissue culture. First application, improved hybrids by somatic hybridization. Now hybridization means production of new varieties of plants. The protoplast fusion we have taken, that is called a hybridization technique. Somatic. The protoplast fusion is also called a somatic hybridization. Okay, so improved hybrids, that means what? They are becoming more and more good quality hybrids will be formed by somatic hybridization. So the second point, somatic hybrids are giving synthetic seeds. When the seeds are produced, they are called synthetic seeds. The synthetic seeds are conservation of biodiversity. The synthetic seeds can successfully germinate and grow. So enormous number of plants will be formed. So new varieties will be distributed in our surrounding, biodiversity. The next application is production of disease resistant plant. Now I already told you, by applying the technique, the plants which are produced are healthy and stronger. So when the plants are healthy and stronger, they cannot be easily affected by disease. So they are called a disease resistant plant by meristem and shoot tip culture. Meristematic culture and the shoot tip culture, disease free plants can be produced. Production of a herbicide resistant plant. Now we have to know what is the meaning of herbicide resistant plant. Now what is the meaning of herbicide? Suppose a farmer is cultivating one type of plant, maybe rice plant, paddy. In the field, unwanted plants will grow. They are called weed. Okay, they are called what? Weed. Previously, they used to remove the weeds and all manually. The workers will be allowed to enter into the field. They will search for unwanted plants and all. They will uproot and throw it away. That is a manual way of removing the weeds. But now, one chemical is available. We have to spray the chemical in the field. What the chemical is doing? It is killing only the unwanted plants. Okay, but it will never do any harm to the crops. Okay, that is called a herbicide. Now that is called what? Herbicide. But side effect will be there. Okay, when it is sprayed, all the weeds will die. But the chemical is also doing some harm to the crop. What is that? Reducing the rate of photosynthesis. So that is called a herbicidal side effect. At the time the crop cannot produce normal grains. So the grain production will be affected. But what we have to do is, by applying the tissue culture, we can produce herbicide resistant plants. That means the plants, the paddy plant, will be resistant to herbicide. Herbicide will kill the weed, but no side effect. The side effect will not cause any problem to the crop. The such plants we can call as herbicidal resistant, heat tolerant. But nowadays you see that because of climatic changes everywhere, it is a global warming is there. The temperature, average temperature is increasing. So the plant should be more and more heat resistant. By this method we can develop heat resistant plants. Even if the temperature is going little beyond the limit, they can bear the temperature. Micro propagation technique to obtain large number of plantlets. Now the micro propagation, this only what I told is callus. We are dividing into 10 or 15 or 20 pieces. Each one will develop into one plantlet. That is called a micro propagation. Enormous number of plantlets will be produced, and the trees also will be produced. What for? For forestry, afforestation. Already forests are destroyed. Okay, deforestation was over. Now we have to bring back that forest. For that, forestry. Okay, that is for forestry, it is very, very useful. Last point, production of secondary metabolite. Already we have seen the tabular column. There are certain byproducts by this technique, but the byproducts are not useful for the plant, but useful for mankind. So the secondary metabolite for pharmaceutical means medical or medicine preparing companies, pharmaceutical companies. Cosmetic, all the face creams, lipsticks, everything. For that also secondary metabolites are there. Then food industry, some are used as a food supplement. For that also they are using this technique. Micro propagation of banana. What is the meaning of a micro propagation? Just now I told you the callus should be divided into enormous number of pieces. Each piece will develop into one plant. Many plants will be produced. Now for banana, what is that? Maintenance of high standard of homogeneity. Now the quality of the banana will be high standard. Okay, good quality, highest we can say, highest quality. And also homogeneity in the field, same variety of plants will be formed. That is called a homogeneity, genetically similar plants. If one plant is having high standard, all the plants present in the field will be high standard. So maintenance of high standard of homogeneity in pineapple, okay, banana, strawberry and potato. Now all these things are food products, that is plants for us. So we can maintain the high quality of pineapple, banana, strawberry and potato. Now we will start with artificial or synthetic seeds. In the previous class I told about what is the meaning of synthetic seeds. Suppose we are following plant tissue culture. At the end of the technique we are finding the plantlets. The plantlets are developed by plant tissue culture. Now after hardening, the plantlets are taken to the natural environmental condition, that is in the nursery or in the farm or in the garden. Okay, the small plantlets are growing bigger, they attain maturity, they start producing flowers, fruits, and inside the fruit the seeds are formed. The seeds formed by the plantlets developed after tissue culture are called the synthetic seeds. The seeds produced by the plants produced after tissue culture are called the synthetic seeds. They are also called the artificial seeds because the seeds are not came from the plant which developed naturally. The plant developed artificially. So that we are calling the seeds also as artificial seeds. Now we'll take what is the meaning of artificial seeds or synthetic seeds. Somatic embryoids. Embryoids, I already told you, the callus we are dividing and then we are keeping them in culture medium. Then they develop into small embryo. The embryos are called embryoids, somatic embryoids. Produce, that means later on, after maturing and all, they produce synthetic seeds. Then we are coming to the next point. Also, that means synthetic cells also develop from single cell. Instead of taking one group of cells as a inoculum, we can take even only one cell. After that, the cell is developed into mass of cells. That is called a callus. Now the callus, the cells in the callus are with a dense cytoplasm. The cytoplasm is very, very concentrated. Large nucleus will be there. Starch granules will be there in the cytoplasm. Not only starch granules, proteins, oils. Oils means fat, etc. Everything will be there in the cytoplasm. Then different inert materials. Inert means inactive. There are different inactive materials. Chemicals are present like agarose, name of the chemical, agarose. Then sodium alginate. But at present they are inert. Inert means inactive. But when it is needed, they can be used as a substrate. So alginate are used for coating. Coating means covering the somatic embryoids. The somatic embryoids, the embryos will be well protected by these chemicals or agarose and sodium alginate. Then advantages of artificial seeds. What are the advantages we have to see? Artificial seeds, otherwise called synthetic seeds. We have to take the advantages. Millions of artificial seeds. That means we are not developing a few seeds. Millions and millions of artificial seeds can be produced at any time at low cost. That means the expense is very, very low, but we can produce millions of artificial seeds. They provide any easy method to produce genetically engineered plant. Now we have to see what is the meaning of genetically engineered plant. One plant is there. Now the plant is having its own characters. Imagine that it is having about 100 characters. The 100 characters are governed by genes. All the characters are original characters of the plant. Now what is the meaning of genetically engineered plant? We have to see. Now we have to take the genes which are responsible for the characters. A few genes, not all the genes. Some genes must be altered. Some genes must be altered in order to increase the power of the character. In order to increase the power of the character. For example, one plant we are taking, one crop, maybe paddy plant or groundnut or anything, a crop plant. Now it is having many genes, but one gene I am taking. The gene is determining the number of seeds produced by the plant. It is only determining. So what we are doing, we are selecting the gene and we are altering the gene. So the power of the gene is increased, as a result of which the plant is able to produce enormous number of seeds. Not usual number, it may be double or it may be even triple. That is called genetically engineered, means genetically modified plants with desirable traits. Traits means characters. So the character what is developed after changing the gene should be desirable. It means it should be beneficial. According to our wish, the character must be changed. That is called genetically engineered plants with a desirable trait. No engineer, no scientist will change the gene to give bad impact or bad result. They will always try to develop the better variety and better quality. Further 20, this point is given. It is easy to test the genotype of the plant. What is the meaning of genotype? Genetic composition. Now, what type of genes are present in the plant? We have to see. That is called the genotype of the plants. They can potentially stored for long time. Now, such seeds, we are taking artificial seeds. What the seeds? They can be stored for long period. Now, generally, the normal seeds and all, if you take, they have their own period of storage. Maybe 6 months, 1 year or 2 years. After that, automatically, the seeds will be spoiled. Now they have their own period of time. But if we collect the artificial seeds, they can be stored for long period of time, maybe 5 years, 6 years, 10 years, 12 years, like that. So under cryopreservation method. Now already we have seen the cryopreservation. Once again we will study about it. So the cryopreservation is one technique. By that method, the seeds can be stored for any longer period, 10 years, 12 years, like that. Artificial seeds produce identical plants. That means genetically identical plants. All the plants will be having the same characters. The period of dormancy of artificial seeds. What is the meaning of dormancy? The period of dormancy. Dormancy means the inactive condition but good condition. For example, we are taking some seeds, general seeds, we are drying it and we are storing it. Now it is not germinating. That means we are keeping them in the storage. That is called a dormancy period. But whenever we are sowing the seeds in the wet soil, then the seed will become active and it will produce the young plant. That is called a germination, sprouting, we are telling. But here, that before sprouting, the period of storage, we can call it as dormant stage. The period of dormancy of artificial seed is greatly reduced, hence the growth is faster. Sometime, now we are storing the seeds, but we want to that is make the plant to develop, but it won't take the season and all. No season, nothing. Once we start germinating them, immediately they are germinating and growing very faster within a shortened life cycle. Not only that, the life cycle of the plant will be reduced. For example, the normal natural plants life period is 3 months or 4 months. But if you take the artificial seeds, once they start sprouting or germinating, the life will be only 75% or 60% of the total life. Before that, it is growing well and it is producing the product, and then it is completing the life cycle. So these are all benefits, or these are all the advantages of artificial seeds.